All right, good morning, everyone. I hope everyone's been doing well among these crazy times. I know for future classes, you guys are probably not going to be in this predicament, but for us right now, we're at home and we're doing online lecturing. So, this week's lecture is going to be on animal development. The last two lectures that we've had was talking about the process of meiosis and the process of genetic variation and the Punnett square and how do parents pass on their genetic trait to the child. So, with that being said, now we're going to combine the two and understand how a child gets developed, or how an offspring gets developed, or how a puppy or a newly fungal spore is going to develop. And this is all based on right after the process of fertilization, where we have two gametic haploid cells fusing into one zygote, forming a diploid cell, and with that singular diploid cell, how does this form into a whole organism? So let's just get started. Uh, yeah, let's just get started. So, so right here, as I said, we're starting with two gametic cells. One's a sperm, one's an egg, and after the process of fertilization, now they become a zygote. That's a fusion of both the egg and the sperm cell. Now, just like mitosis, a zygote is a singular cell. For it to become a multicellular organism, it needs to undergo a rapid change uh, of growth, or rapid growth, so to speak. And so the thing is, how does a single cell, fertilized cell, diploid cell, become multicellular? It has to undergo mitosis. Only mitosis, not meiosis. It will undergo mitosis. So the zygote undergo mitosis and start creating a whole, an embryonic ball, so to speak. And it's exponentially growing because one cell divides into two cells, and each of those cells undergoes another process of mitosis, which is four, and then the 4 goes to 8, 8 to 16, and it just increases exponentially after that point. And so, and just like mitosis, right before the cell splits into two pieces. So during that telophase, okay, I'm recalling a lot of past terms because this is just a review for you guys, hopefully. Right as we hit the telophase mark, remember the two nucleuses form on both ends. And then in order to separate the two nucleuses from each other, it needs to form something during the process of cytokinesis. And this is what we call a cleavage furrow. Cleavage just means it starts to pinch. Okay, so for a zygote to form multiple cells by undergoing mitosis, it has to undergo a process called cleavage. And that all it means is that it's going to start splitting apart rapidly, forming an embryonic ball. Now, once it forms that embryonic ball, now we got to assign each cell a specific role, so to speak. Because right now they're all in the process of stem cells. They're a blank cell. They don't know what type of cell they want to become. Right now they're just a stem cell. We don't know if they want to be a heart cell, a brain cell, a muscle cell, a skin cell. So it has to undergo a process called cell determination, and that's the only way that we can differentiate the difference between which cell goes to what. So the cell undergoes changes in the gene expression, and so it depends on the time and place in the development of the embryo, hence the differentiation. So genes play a huge role. Remember, the central dogma back in during exam 2, we talked about the central dogma, DNA to RNA to protein. The whole point is to create proteins so that the proteins help develop the cell, gives it its physical characteristic. Okay, depending on what protein is being developed will determine what cell it becomes. And the determination is the process of the commitment of the cell line to a particular developmental pathway. So it could be a neurological cells. And the cells that do not change, again, these are what we call the embryonic stem cells. Okay, there are two different types of stem cells. The one that we're talking about is embryonic. The other one, it's the adult form, which we may talk about later. So development in animals begin with the formation of zygote. I'm reiterating for a reason, so hopefully it's making more sense. Once the zygote starts to divide, it's called the embryo. Okay. Now there are four-ish phases for early embryonic development. The first one is cleavage. Now remember, what is cleavage? Cleavage is just the rapid division of the zygotic cell or zygote. Because remember, the zygote is just a single fertilized cell. Let me repeat that. A zygote is a single fertilized cell. Once it starts dividing rapidly, that's when it becomes an embryonic cell or embryo, so to speak. Okay. The second step. So that's step 1 is cleavage. Step 2 is gastrulation. Step 3 is neurulation. And step 4 is organogenesis. Now, let's just look at the words right now. Okay. This is where breaking down the words might help. Okay. Say for on the exam, you're looking at these words right here, and I'm asking you to define it. How would you define it? Say that you studied so much, but then you had a brain fart. Let's just look at the word. So if I look at the word right here, gastrulation, let me get my pointer. If I look at gastrulation, gastro sounds something to do with the digestion, something. So I don't fully know what this word means right now. Okay, I'm gonna skip it. And this is a good test taking strategy. If you don't know a word, skip it, go on to the next choice to see if you can figure out what that word is. And most likely on a multiple choice, it's probably going to have a definition, be like, oh, what process is a formation of multiple cells from a single cell or something like that. I don't know. And you're supposed to pick A, B, C, or D. Again, this is a good way to break the words down. So gastrulation, say that we don't know. I don't know right off the top of my head right now. All right. I'm going to go to the next word. Well, cleavage, again, cleavage furrow, that's how I would see number 1. Number 2, gastrulation, I have no idea. Number 3, neurulation, neuro. Okay, neuro sounds like neurons, nerves, brain. Okay, so I'm thinking step 3 is the development of nerve pathways, maybe, because again, it's an embryonic cell. They don't have nerve cells yet. They don't have a pathway. So neurulation might be the development of the brain or something. Okay. I see that has something to do with the nervous system. Number 1 has something to do with the division of cells. Number 2, I have no idea. Number 3 has something with nerves. Okay. So step 3 is most likely going to be something with nerves. Oh, step 4, organogenesis. Well, organ. Number 4 is organ. Okay, and genesis, genesis is the creation of. So organogenesis is probably the creation of specific organ or organ systems. So that's how I'm going to see it. So step 1 is dividing the cells. Step 2, gastrulation. Maybe it's forming the digestive tract, esophageal tube, or something gastrointestinal. That sounds like digestion. Neurulation sounds like nerves, and organogenesis is like the rest of the organs. That's what I'm thinking. Okay, so that's how you would break down these words. So let's explain the first step, the process of cleavage. Now, this is a great visual representation. Like I said, zygote is just a fertilized egg cell. Once the sperm penetrates the egg and injects the dad's DNA into the female's egg, now it becomes a fertilized zygote. Okay, and then the process of cell division begins. So the first phase is called cleavage, which produces a ball of cells. And this is what we call a blastula. Okay, not blastoma. Blastoma is something totally different. So blastula with an interior liquid-filled cavity. So there's going to be a lot of water inside, a lot of different nutrients inside. So cell division begins. A morula, which is the, I believe it's about 8 cells, is the stage right after the first cell division. And then blastula is now the big ball of multiple cells. Now, notice right here on the blastula, there are 2 locations called the animal pole and the vegetal pole. We'll talk a little bit more about that, I believe. But just know that in the animal pole, you're going to have a region called the blastocele, something like that. And we'll talk a little bit more about that. Okay. So again, this is just a great visual representation of what happens inside the womb as the fertilized not gamete, but the fertilized zygote cell begins to divide. The second phase is called gastrulation, in which the blastula cells rearrange themselves into the three layers that give rise to the specific organ and tissue. So there's going to be three specific types of cell during the process of gastrulation. You might have heard these three specific types of cells before. It's going to be called the ectoderm, the endoderm, and the mesoderm. Those are the three different types. Let's just look at the visual representation right here. So right here, notice your blastula. You're going to have your animal pole on the northern facing point of the embryo, the vegetative pole, which is on the southern point of the embryo, and the blastosal. I don't know how to pronounce it. Bear with me. So now we have a mesenchyme cell prompted vegetative pole to pinch inward towards the animal pole. So on the vegetative pole, what's going to happen is that they're going to start forming a little pore, and they're going to increase that pore so that it forms, as we can see, a tube-like structure. And this tube-like structure is our digestive tract, believe it or not. It's the first development of an esophagus down to the stomach, down to the intestine. All those cells will eventually form into those specific organs, but in a very simplistic development, in the beginning of the development, because the cells are not defined just yet, they're just drawing a rough blueprint. They're going to be like, hey, here's the mouth, here's the butt, here's the digestive tract. As we develop into a more complicated organism, that's when we'll rearrange ourselves. But here, let's get the blueprint down. Okay, it's like developing a house. You want to have the front door going in and the back door going out, but you're not gonna, overly develop the living room area and decorate it just yet. You need the blueprint first. You need to know how we're gonna go in, how we're gonna go out. Okay, so the mesenchyme cells are those cells on the vegetal side of the embryo that will intrude or implode, not implode, but will just pinch inward all the way up to the animal facing or animal pole to create that digestive tract. And as you can see, as it creates, the mouth of the organism is going to form on the animal pole, and the anus, the butt, will be forming on the vegetal pole. Okay, an archenteron elongates towards the animal. This is the only way for the digestive system to be pulled towards the animal pole end. It needs to latch on to like hooks, so to speak. The archenteron, I guess, is the, what do you call it? Oh, actually, sorry. The hook pieces are the mesenchyme. The archenteron is the inward protrusion. So it's what we call the inward pinching cells that are going in to develop the digestive tract. Okay, and the three tissue layers form again: the ectoderm, the endoderm, and the mesoderm. Now, ectoplasm or ectocytosis, that means outside. Inside means inside. Meso sounds like a middle. Middle, muscles type of cell. So again, derm just means skin or layer, or essentially layer. So ectoderm is your outermost layer, endoderm is the innermost layer, and the mesoderm is the middle layer. Now there is a thing called the epiderm, but the epiderm means the top layer of your skin. We're not going to get into that. Just know that there's three different types of cells that are forming. Again, these are the main types of cells that the endoderm, mesoderm, and ectoderm will soon develop as the development goes underway. So endoderm, again, it's the inside, it's the smooth cells. That's what usually creates our lungs, our pancreas, our digestive system. Again, it's forming the digestive tract, the bladder, some hormonal glands. That's all endoderm. The endoderm, that's their role. Mesoderm, again, it's like muscles or something like that. It's going to make more of the body mass of it's going to take up more mass of the cells of the embryo itself. And that's what we use to develop the blood vessel, the blood, the sperm and egg, even the kidneys, the adrenal glands, the bone marrow, muscle, and skeletal tissue. So that's all the mesoderm. It's the middle meaty part of our body. Okay, and the ectoderm is like our eyes, our teeth, our epidermal layer outside. Anything that's you can see on the outside is generally going to be made out of our ectodermal cells. Okay, so right here, gastrulation and the echinoderms, the vegetable pole or vegetal pole invaginates and meets the opposing wall, obliterating the blastose cell. That just means that it's just going to go inward and then just create that digestive tract. It's the newly formed cavity. The archenteron is the cavity itself that's being formed right here. So that's essentially the airspace in the esophageal area. And the blastopore is the opening of the archenteron that becomes the butt or the anus of the new cell. Okay, and these are a lot of terminologies. So again, if you are confused a little bit, look through the slides or replay several seconds, several minutes back, and then just go through the explanation again. Now, flay of a blastopore. Again, the blastopore is the butt end. So you're going to have the protozome, which is organism whose blastopore becomes the mouth, or you can have a deuterostome, where the organism whose blastopore becomes the anus. So it depends on the organism itself. Don't ask me why, I don't know. So just know that there's two fates that could happen. So right when that pore forms, as the archenteron is going towards the animal pole, that's the vegetal pole that has that little anus forming, that could eventually become a mouth or an anus. And if it becomes a mouth, that's a protosome. If it becomes an anus, that's a deuterostome. Okay? And this is just a different process. Again, you guys have the slides. You can go through it and just look at the process here. Now, neurulation is the third step. Okay? Again, neurulation sounds like nerves. The nervous system is the first organ system to develop. Again, we need a nervous system, especially that we are multicellular, complex, eukaryotic organism. We need a control center. If we start developing the heart first, or the stomach first, or the the lymphatic system first, without the nerves to control the heartbeat, you're going to develop a heart for no reason. So you need that nervous system to at least send those electrical signals out and really tell the cells what to do. You need to create a management system, so to speak, before even running a business. You need to have that management system in place. How are you going to manage, so to speak? That's an analogy. So, okay, and this overlaps a primitive streak formation. Just means that it overlaps a very primitive, basic underlying form of development, because again, bacterial cells and archaic cells do not have a nervous system. So all they do is they divide and they know what to do. But because we're more complex, the nervous system needs to override that type of formation. So once the nervous system is in place, the rest of the organs can develop. Again, we created the control center. We have a management system that says, hey, you go develop your brain. Actually, the brain's ready to develop. You go develop the heart cell. I want you to develop into muscle cells. We need you to become a skeletal system. And so now we have an efficient way to delegate and distribute tasks to different cells so that they can form into those multiple cells. And so the notochord sounds very similar to the spinal cord. The notochord grows and induces overlying ectoderm to form the neural plate. Okay, so you can see here's the neural plate. It's gonna start pinching, sort of forming this little plate-like structure, and then the nervous system cord is going to be right underneath that little dip layer right here. So right here, just reading off the slide, thickening and elongation ectoderm over the notochord thickens to form a neural plate, which elongates adjacent regions, will form a neural crest and an epidermis. So again, we have the most of our nerves, especially on our fingers, are right underneath the skin. That's why when we touch something hot, our brain is receiving signals on the surface of our skin saying, hey, ouch, 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 is really hot. Or if I got a pen and I start stabbing myself, you're going to feel that because a lot of your nervous system, your nerve endings, are right underneath the layer of the skin, the epidermal skin. This is how we have those touch sensation. Now, granted, our fingers and our toes have more of a touch sensation than the rest of our bodies. If you were to touch the back of your neck, you're not gonna, feel as many. You're not gonna have as many receptors to feel it because we don't touch with the back of our necks. We touch more with our digits. So cells of a neural plate fold to form a neural groove, and the surrounding neural folds. Again, right as you see right here, we're forming that tube-like structure. This is where the nerve cells are going to run through this railway track. It's like a power line, okay? If you ever look at power lines are always circular, but if you were to cut one in half, you're going to see multiple wires inside a power line. Our body has the same type of process. We have a little power line, we're going to fold it together to forming a hollow tube, and then the nerve cells is going to form inside. Okay, and you can see that the dorsal surface, or the back end of the surface, is going to start forming until it fuses together. So the neural folds will fuse, forming a neural tube, and the anterior portion forms the brain, and the rest will form the spinal cord. So we're going to have one end of it forming the brain itself. That's how our brain forms, and the rest is the spine. Okay, so the neural crest is a critical structure that guides formation of several organ systems. Again, we need to have this structure. So the neural crest forms on either side of the points of fusion, migrates to other regions of the embryo, and it's the destination to many different types of cells, or it helps differentiate. Okay, so right here you can see the neural crest is right here. As it closes off and it pinches away, these little neural crest cells have instructions. This is like my managing system. I'm the CEO. I want to talk to all my managers. I'm going to say, hey, I want you to do this, do this, do this, do this. So each individual manager will have a role, and say that one manager's specific role is to create the heart cell. So he's going to go out to the rest of the body, and then he's going to tell the cells, again, whatever cells we're looking at, the mesodermal cells, hey, we need to create a heart system. Let's go. Let's create. Okay. That's essentially what neural crest cells are. They're little managers with a specific task that I assign to them. And to be like, hey, we need to develop this now. Go out. So right when the neural tube closes, the neural crest cells are pinched out, and so to speak, it's going to get dispersed throughout. And you can see right here, as it pinches, the neural crest cells are going to disperse out, which is what's happening right here to the rest of the embryonic cell, or embryo. Okay. So now we're going to have blocks of mesoderm called somites, and this is what forms the outside of the neural tube. Again, we need to protect this inner tubing. If we don't protect it and we accidentally damage it or chop it, that's when now the nerves are no longer communicating with each other. It's essentially like a roadblock. So if you can't send brain signals to the rest of your limbs, this is where paralysis happens. Because now the nerve endings are cut. It's like taking a wire from a lamp and you're cutting the wire. The power can't get through anymore. The lamp doesn't work. Until you're able to reconnect those wiring, will the lamp then work? And this is a whole developmental process today where we're trying to discover a way to reconnect all these multi-millions of nerve cells, because there's a lot of people who are quadriplegic or have paralysis through their body, and it's because of a nerve damage. And there's talk going on where we're taking stem cells and injecting them into the spinal area or the nerve area to help hopefully repair and reconnect the cell. But there's a lot of litigation that goes on with that, and we have to go through clinical trials and so far. So it's a very slow process as of this point. That's just extra information. So now going back, becomes the vertebrae and associate parts of the segmented body, the vertebrae, the muscle, kidney, reproductive structure, heart, and blood vessel. Again, we need these somites to protect all of that. The third step is organogenesis. I thought that was the fourth step, but it's the third step. The third step is organogenesis, in which the organs begins to form. Again, we start with the embryo near the end of gastrulation. Okay, notochords induced is being developed, as you can see right here. Now we're creating the notochords, the neural folds, and then now we create, this is, you can see right here, now the spinal cord, or the spinal area, is now being formed, and the neural crest is now going to be distributed to the rest of the blastula. Okay. So early embryonic development, the first product of cleavage is a tightly packed ball called the morula. Again, that's when the singular zygote divides into 4 cells and into 8. This is what we call the morula. And then after the morula is then rearranged itself into a structure composed of 1 or more layers of cells, so like an onion, so to speak. And then the inside is mainly water. This is what we call the blastula. So the blastula has liquid filled within it and multiple layers of cell surrounding it, just like an onion. Morula are just cells. There is no liquid found between them. And the interior cavity is called the blastocell. Again, we're repeating a lot because this is a lot of information, and hopefully as I'm repeating it and as I'm introducing it to you in a different way, that this all makes some sense. Gastrulation produces embryo composed of 3 layers of cell: the endoderm, the mesoderm, and the ectoderm. Again, read through this if you must. Okay, and then we're going to develop the spinal cord, the notochord, and it induces development of the ectodermal tissue that lies right above it. And right when we form that neural tube, the neural crest cells is going to be dispersed. Okay, so I had to pause really quick, and now I'm unpaused. And so this is, again, just a review of the early embryonic development. Okay. Now groups of neural crests again spreads out, and now each of those managers are now going to go to the specific part of the embryo saying, hey, we need to develop this system. You got to go develop the skeletal system. You got to go develop the heart system. I need you to develop kidneys. I need you to develop the lungs. Okay. And there's an animation, which I don't have right now. So a small group of, so early stages of development, it goes from general to specific task. A small group of superficially similar cells give rise to 3 different layers of cells, which give rise to tissues and then gives rise to organs. Again, we go from a general development, and then as we become more and more complex, now we need to assign specific role. It's like me opening up a lemonade stand. Bear with me with this analogy. So if I was going to open up a lemonade stand, I am the owner, I'm the manager, I'm the worker, I'm the buyer and the seller, right? I'm everything. But as my lemonade stand grows, I need to hire more workers because I cannot take the task all on my own anymore. So now I'm going to hire people to work as a cashier. I'm going to hire people to now make the lemonade itself. So, and I'm going to have people who are going to go out to the markets to buy the lemons, to bring it to me, and to buy the lemons and the sugar. So I need the producers to come here to give me the stuff. So now as we get bigger, I need to assign more tasks because I can't do it alone anymore. That's essentially how the cells think. Once they get too big, they need to delegate responsibility. And again, a business is like an organism. So what factors underlie development? Induction is a process in which some embryonic cells direct the development of other embryonic cells. What does that mean? It means that, okay, once I give you a task, now they are going to tell the other cells to do another task. Essentially, it's a passing of tasks or passing of instruction. Okay, and as you develop, it's going to be highly regulated by the genes and the proteins. Again, this goes back to chapter, I forgot what chapter was, 7, where we're talking about the central dogma, DNA to RNA to protein. Again, that is the process at which all cells are developed. We need our DNA, and as long as our DNA is intact and not mutated, our genes are going to be developed through the RNA transcription, and then from RNA to protein with the translation, now we can start developing the protein. And depending on what protein we are creating will determine what type of cell comes out. Okay, and again, in science, in order to see this process, we use an organism called a fruit fly because it's one of the most simplistic organisms that we can find, and it's what we base a lot of our scientific study on. Drosophila melanogaster. I call it the Drosophila fly. I used to call it Drosophila. Side note. Okay. And it's a critical early step in development. Again, you need to understand the positioning of where the cells are. We have an anterior position of the cell and a posterior position of the cell. And depending on which side you're on will determine does this form the head or does this form the butt. So it really depends on the poles. Okay. So positional information in the Drosophila fly is provided in part by a protein called the bicoid protein. Again, depending on where the bicoids are, that's what forms the thoracic compartment of the fly, or the head compartment, or the butt compartment. Okay. Greatest concentration is at the anterior end. So if I have a whole ton of bicoid protein in the fly itself at the anterior end, that's what's going to form the head of the fly. Everything else is just going to be programmed to, okay, here we're going to form the butt. And this is a process of regulation. Again, remember, back in chapter 7, we talked about the lac operon, where if lactose was present, the operon will then work. And if lactose was absent, the bacteria cell is going to have a repressor protein latched into it. You can think of the bicoid like a process of that, where if we don't have bicoid, there's a repressor protein that prevents the rest of the body from forming the head. But once the bicoid protein is available, now the repressor is it's latched off. That gene is turned on. Now the gene's turned on, saying, "Hey, this is the gene. We're forming the head. This is the head gene." And so that's how the head will start developing. Again, this all comes into the process of protein regulation. Woo! Okay. Now, a morphogen, a diffusible substance whose local concentration affects the course. Again, I'm reading off the slide. Whose local concentration affects the course of local development in the organism. Let me see if I can explain this in a more simplistic way. So morphogen essentially is is based on the concentration. Again, it's the bicoid. We're still talking about the fly. So morphogen, depending on how much a concentration is throughout the cell area, this is what we call a morphogen. And as you diffuse this protein throughout the rest of the cell, depending on the concentration, it will determine whether this is going to be the head, the butt, and the thorax or abdomen. And you can see right here, originally, a lot of the bicoid protein is concentrated on the anterior end, but the bicoid protein is known as a morphogen because depending on the concentration of this protein will determine, again, what structure is being developed. It's just morphogen is a general characteristic of a specific protein that affects the development of future physiological structures. Okay, simplistically speaking. So, an analogy of that would probably be, say that I don't know. Say that I'm gonna give a very rough example, so bear with me. Say that I'm a bread store owner and I make a whole ton of bread. Like I make so much bread in downtown LA, but and the reason why I make so much bread in downtown LA is because I know a lot of people like to eat bread in LA. But as I diffuse my product over a greater area, say that we're going from LA down to Costa Mesa, not a lot of people are going to eat my bread because it's so far away. And so they're not going to have a strong concentration there. It's not going to have a strong bread culture there. They're probably going to be more into the steak culture because there's a steakhouse there. That's just a very rough representation. I can't think of another analogy off the top of my head as of this point. So just bear with it. Okay. So in general, morphogen diffuses from one end of the cell to the other. Binding with these latter cell morphogens prompt the production of transcription factors. Again, DNA to RNA needs transcription factors to work. Without the transcription factors, transcription will not work efficiently or will not work at all. So this will help regulate which genes are going to be turned on and off. And so all morphogen work through the kind of concentration gradient, and there's multiple different types of morphogen. But in the Drosophila fly, the morphogen is the bicoid protein. Okay, and the developmental works goes through a genetic cascade effect. Again, we all do cell signaling, and so as different proteins are being developed, and as the cell receives that signal, it's going to cascade down, turning on more or less genes. Okay, and this cascade again affects on how the development proceeds. So if in the early embryonic stages, if there's a problem with the signaling of the cells, this is where deformation occurs in the embryonic development. So the cells need to be happy, and if there's a problem, deformation or possibly apoptosis will occur, killing off the embryo. And this is why sometimes, you know, depending on the developmental stages of an organism, if the mother organism is ingesting a lot of different chemicals and pesticides and herbicides, that might affect the developmental process of the embryo, because those chemicals affect the cell signals inside while the embryo is developing. And if you are to cut off that cell signal, deformation occurs or possibly death. Okay. Subsequent levels of genetic instruction is specific to the development of the different body section, and the control is exercised over the developmental specific structures such as organs. Okay. Whew, man, that's a lot of information we went through. I'm pretty pooped out. This is a 36-minute recording so far. I'm looking on my recording right here, so it's a long. We don't have that many more slides left, so bear with it. We have about, I think, 10 to 15 minutes left. That's it. So hopefully we can get through this and you guys can be on your way. So within the many developmental genes of the animals, different flies, mice, and humans, there's about 180 DNA bases that are identical between all of our multicellular organisms. And this is what we call the homeo box. This is a specific region of the DNA that is extremely conserved. This is found in, like we just said, mice, rabbits, humans, dogs, cats. And it is believed that if anything was to change within this homeo box, instructions, the base instructions of development will totally be annihilated. Not annihilated, but mutated to the point where mutations and deformations will occur. So we need this to be extremely safe, extremely conserved, extremely identical for us to have the same process. And so scientists are now looking into the differences in the homeobox genes and their protein as a mean to learning more about the animal and human development. Yes. Okay. And as you can see, at a very young embryonic developmental stage, if you look at all the organisms right here, from birds, bats, dogs, humans, primates, you name it, we all have a very similar developmental shape. You can see that here is the anterior end where the head is probably going to be found. On the bottom is the tail end where the butt or the feet or the tail is going to form. And you can see that we all have a very, very, very similar type of development. And again, this all goes into that homeobox gene, which is highly conserved among all organisms, or most organisms, eukaryotic organisms, I should say. And so you can see just the developmental process is it's nearly identical to each other, okay, in terms of embryonic development. All right, and so apoptosis, we need apoptosis. Again, I talked about during mitosis, the chapter 8, that we are born with web-like hands. Now, chances are most of us will not have a big webbing between our fingers. And how does the body know to get rid of it, or how do we even get rid of it? We're not going to cut the webs off of our finger with scissors, okay? That's a little too painful. But we have a process called apoptosis, and it's a programming in the cell that says, hey, this is extra information. We don't need this. This is a waste of energy. We know that we're going to develop into a primate which is living on land. We don't need the webbing to swim anymore. So why not get rid of that excess uh, membrane, so to speak, and redistribute the materials in that excess membrane to the rest of the embryos, so that we can use that to at a more efficient level. Excuse me. And so that's the whole process of apoptosis. It's just a means to kill off the cell, recycle those proteins, DNA material within that cell, and redistribute to the rest of the body. Especially that we know that we're not going to be swimming. We're not amphibians, and we're not aquatic creatures. Okay? And development is something that is thought of as an ending of birth, but it continues through the life of an organism. Again, just look at us, for example. We have toddlers, then we have children, and then adolescents, and then teens, and boom. For some reason, right when you go from adolescent, or adolescent and teenager is probably about the same thing, but right when you go from child to teenager, somehow they grow a feet or two within that time frame. And again, that's the developmental process. At a younger age, they still have molding capabilities in their bone structure. It's not completely solidified just yet. That's why when they're younger, they can heal much faster, and they're able to grow. And that's an example of puberty and menopause. Oh, and we're done. So that was nice. I didn't even know we were done. So that was another 4 minutes, guys. Let's just go back. So developmental happens all the way until menopausal state, where our hormones are finally at a very stable level. We don't need to develop anymore. And this goes back to the caloric intake. Remember, at a younger age, because we're still growing, because we're still developing, we need a lot more calories and a lot more nutrients. And if we don't have a lot of nutrients or calories at a younger age, chances are this is what's going to lead to a shortness in height at a later age, less developed muscle structure, health problems later on. It could diet is a huge role. But remember, once you're finally fully developed, that means you're no longer going through puberty. The amount of calories that our body needs drastically decreases, because we're no longer using that excess energy to grow. Now we're in the process of maintaining our body itself. So if you found yourself eating, say, 4,000 calories as a teenager, and during that time you never got chunky, fat, or big, it's because your body was still using all that energy. You were much more active, you had all that energy, and it's using it to grow and develop the rest of your body. But probably around the age of 21 to 25, your body stopped growing, but you kept that, maintained that specific diet. But now, as you see, as people get older, myself included, we get a little chunkier. And it's because our metabolism is slowing down. We're no longer growing anymore. And so that's just a good indicator saying, hey, we're no longer growing. We're in the maintaining phase, so let's cut back on the calories. Again, it's hard for all of us, myself included, and so that's why rigorous workout to keep up with the amount of calories I eat is a way to help maintain my body size. And again, that's just an example. It's just extra information for you guys to know. But for the next chapter, we will be talking about more about genetics. Hopefully, this was interesting to you guys. It was for me, and so stay safe out there, guys. Be healthy, and I'll see you guys in the next lecture. So, cheers.